Hello everyone, my name is Anh Pham. I'm an ocean biochemist at UCLA. Today I'm gonna talk about the work that I did at my previous institution at Lotion in Paris with Olivier Aumont, and that is gonna be about zooplankton demand modulates the iron recycling and microbial iron consumption. So over the last few decades, thanks to the geotherapy program and other projects that have cruises going around the world ocean. We have so much iron data, and our knowledge in terms of the ocean iron cycling has significantly improved. We know that iron coming into the ocean from atmospheric deposition from continental shelves and hydrothermal vents and in the interior ocean away from sources. Biological processes, the interaction of biological processes, that is the interaction between the uptake of iron by phytoplankton, the reionization of iron from seeking organic particles, the scavenging of iron and by organic particles and inorganic particles, the desorption of scavenged iron from sinking particles, and the release of ligands, which are organic materials that protect iron from scavenging. All of these biological processes interact with each other to control the ion distribution in the interior ocean. So with the emerging role for biological processes in controlling the ion distribution, now we care more about how iron is going to be uptaken and consumed by first phytoplankton, but we also care how iron is consumed and stimulated by other players in the food chain, in the upper food chain, like for example, zooplankton in fishes. For example, we know that zooplankton consume microbes. That is, they eat bacteria and phytoplankton so that they can indirectly impact the ocean ion cycling by changing the biomass of microbes and changing the microbial ion consumption. But also, they can play a direct role in controlling the ocean ice cycling, which shown by the work by Richard et al. 2020. They showed that the ice recycling flux in the upper ocean mostly performed by zooplankton, and mm, that's how phytoplankton can get their iron to support their photosynthesis. Also, they showed that the iron recycling flux by microzooplankton is a function and it's really sensitive to the food that zooplankton eat, and it's also really sensitive to the iron requirement, the maximum iron storage of microzooplankton. That is the iron stoichiometry of zooplankton. Mm, And also, we know that from current study by Mezzo and Gambrek 2020, we know that zooplankton can also modulate the ion export flux, which shown in the figure here. Here, basically, what it's showing is the ion carbon ratio in fecal palette by zooplankton that is sink from the surface to the deep ocean has a high ion to carbon ratio than the ion carbon ratio in the food that zooplankton eat. So zooplankton can enrich the sinking ion particle sink from the surface to the deep ocean, so they can enrich the sink particular sinking ion. But there's still so much uncertainty and unknown because we lack observations and measurements and understanding of the variability in the iron requirement by zooplankton, their maximum iron storage and their optimum iron requirement. Basically, our understanding the iron stoichiometry iron content of zooplankton is limited by observations, by measurements, and Earth system models that used to project future changes. Still assume a constant ratio for the iron content of zooplankton, which is not supported by what we know from observations and measurements. We know that zooplankton iron stoichiometry can vary as a function of temperature of environment. So my research question are, I would like to understand better the role of zooplankton on ocean ion cycling. The first question going to be, would like to ask is how sensitive the ion recycling and export fluxes is to the zooplankton demand and storage. we can form a simple hypothesis. That is, an increase in the ion demand or maximum storage of the zooplankton will increase the zooplankton ion content and zooplankton ion export flux, but decrease their ion recycling flux, which ultimately decreasing the surface ion concentration. And the second question that I would like to ask is what are the relative role of direct versus indirect impact of zooplankton on ion cycling. When I say about the indirect impact, I'm saying about the impact of zooplankton on microbial biomass and ultimately on microbial ion consumption. And to do that, I'm gonna use an ocean ecosystem model that has been used extensively to study the ocean ion cycling. The model has ammonium nitrate, iron silicate, and represents bacteria, phytoplankton, zooplankton, dissolved organic matter, and particulate organic matter. And we also represent the iron content, the carbon content of bacteria, nanophytoplankton, diatoms, microzooplankton, and mesozooplankton. That is to say that the iron content of all living materials here can vary as a function of their iron requirement, their maximum iron storage, and the iron concentration in the ocean. And to get a sense of how the model works, first I look at the results from the control standard model run. I'm looking at the surface microzooplankton biomass and surface mesozooplankton biomass. So we see that zooplankton biomass is high in the high latitude and low in the low latitude, and also zooplankton biomass can be high along the coast. Following the pattern of zooplankton biomass, the iron content in zooplankton also high in high latitude, southern oceans, North Atlantic, North Pacific, and low in low latitude ocean, and also the values of surface iron photons can be high along the coast. And then I'm looking at the ion recycling flux performed by microbial zooplankton and mesozooplankton. And in the figure on the left here, comparing the figure on the left, microzooplankton with figure on the right of mesozooplankton, we see that the ion recycling flux is performed in the surface ocean, mostly by microzooplankton. The result here is consistent with what we know from some observations and from the results of Richard et al. Modeling study in 2020. And the same thing can be said for the pattern of export flux in the surface. We see that ion export flux in the surface is mostly coming from microzooplankton, where there is a caveat here that we have to keep in mind is in the surface ocean, though maybe microzooplankton play a bigger role than mesozooplankton, but in the deep ocean, the story can be different when mesozooplankton can play a bigger role. So to answer the two questions that I brought up at the beginning, I'm gonna run 3 sensitivity model simulations. Basically, what I did here is I'm increasing the minimum requirement or decreasing the maximum storage of iron from microzooplankton, and I'm increasing the minimum requirement for iron of mesozooplankton. It's going to be clearer when I looking at the results of all the experiments. So first of all, I'm looking at the changes in the recycling flux performed by microzooplankton when we change the minimum iron requirement of micro mesozooplankton, or we decrease the maximum iron storage of mesozooplankton. We see that recycling flux of iron performed by microzooplankton increases when maximum iron storage from microzooplankton decreases, which is consistent with the hypothesis we formed at the beginning. That is, when we decrease the iron content in microzooplankton, their recycling iron flux gonna increase. But on the left hand side figure, on the far left hand side figure, we see that changes in recycling iron flux are not uniform when the minimum iron requirement of microzooplankton increases, there are some regions of decreasing, which is consistent with our hypothesis, but there are also regions increasing. So there must be something else going on here that we have to explain. The same pattern can be seen also in the ion export flux. The changes in, we see that on the far left hand side, again, the changes in ion export flux are not uniform when the minimum ion requirement of microzooplankton increases. We see some regions of decreasing, but we also see some regions of increasing as well. So the patterns are but a little bit touchy, and we have to explain that. So my point here is the changes in recycling flux and export flux of ion by microzooplankton when their minimum ion requirement increases. It's not simple, and it's not really a simple function of their ion content. And finally, we're looking at the changes in surface dissolved ion concentration. We see that even though there's some patterns and the changes in the recycling flux of ion and export flux of ion are not uniform, but surface dissolved ion decreases everywhere when minimum ion requirement of microzooplankton increases, which is consistent with our hypothesis. And surface ion increases when maximum ion storage of microzooplankton decreases, and which saying that these changes seem to be not a simple function of changes in the recycling and export flux, and then we have to explain why that is the case. And we can explain the changes here by looking at the changes in surface microzooplankton biomass. So basically, when we on the far left hand side, when we increase the minimum ion requirement for microzooplankton, we make microzooplankton to become more ion limited. So that's why microzooplankton biomass decreases. And if we look at the changes in the microzooplankton biomass here, it matches the change that we see in the ion recycling flux and ion export flux by microzooplankton. So the conclusion here is the changes in ion recycling flux and ion export flux by microzooplankton is coming more from the changes in their biomass than coming from their ion content. And to explain the changes in surface dissolved ion concentration, actually we have to look at the changes in chlorophyll phytoplankton biomass or bacterial biomass, because when we increase the microzooplankton ion limitation, we decrease microzooplankton biomass, and the decrease in microzooplankton biomass leads to a decrease in zooplankton grazing pressure on phytoplankton and bacteria, and that is going to lead to an increase in phytoplankton and bacterial biomasses, which is shown here on the far left-hand side figure. And ultimately, that's going to lead to an increase in the microbial ion consumption, that is the ion consumption by bacteria and phytoplankton. So because bacteria and phytoplankton increasing in their biomass, they're going to consume more ion, and that's going to lead to a uniform decrease of surface ion concentration that we see at the beginning. So the change in surface ion concentration actually is a balance between the change in recyclable flux and export flux of ion performed by microzooplankton, but also from the changes in microbial ion consumption. So to conclude my presentation, I have two answers for two questions that I put up at the beginning. How sensitive the ion recycling and export fluxes is to the ion demand and storage of zooplankton? I can answer that changes in zooplankton ion demand lead to changes in the ion recycling export flux, mostly to changes in the zooplankton biomass because of changes in their ion requirement, not directly from the changes in the zooplankton ion content. And the second question, what are the relative role of direct versus indirect impacts? That is the direct impact of zooplankton ion cycling through their recycling and export flux, and the indirect impact is the impact of zooplankton on microbial ion consumption. We can say that both impact play a role, and the changes in surface ion concentration are going to be the balance between these two impacts, but changes in biomass and ion consumption of microbes lead to interesting changes in the ocean ion concentration that is not easily explained if we just look at the ion recycling flux and export flux. So that is, I think, is important because so far in the zooplankton ion interaction community, we mostly talk about how zooplankton can directly impact the ocean ion cycle by changing the recycling flux and export flux. But I want to emphasize also the impact of zooplankton by changing the microbe biomass, and that's how they can change the microbial, ultimately changing the microbial ion consumption. That is kind of like the top-down effect versus the bottom-up effect, and the change in ion concentration, the impact of zooplankton on ions concentration going to be both the top-down and balance between top-down and bottom-up effect. And if we that's why we need more data from the field about the zooplankton ion demand and their ion requirement, how they assimilate ion and digest ion in the cells, and data from biochemistry program like geotraces and biogeoscape going to be invaluable to moving the field forward. Thank you for listening. I am happy to take any question.